Good to proceed.
Okay. So is my screen visible, guys?
Not at sorry. Yeah, now
Is
it's
it
good.
visible?
Yeah.
Okay. So, uh, so in the last previous sessions, we basically covered the entire RNA-seq workflow. So now, today we are going to first revise the entire workflow. I'm going to sum it up for you guys. And after that, we are going to move to the project allocations. So you guys already know in which group you are. So accordingly, a project will be allocated and you have to work with the project and replicate the entire pipeline. So for the projects, first of all, you have to execute the entire workflow. That is, you have to complete the entire RNA-seq workflow using the data sets that you're going to obtain from GEO and SRA. After that, you're going to do the analysis and you're going to do the differential expression. That is, you're going to check how the genes are differentially expressed in both the control and the experimental conditions. And after that, you're going to biologically interpret whatever changes have happened and whatever perturbations or any environmental stress or any developmental transitions that is happening, which is causing that particular change in the expression level. This particular analysis will help you gain your skills and you will be able to gain a hands-on experience on the modern RNA-seq data analysis, both tools and workflow. So now to before I start, first we'll revise whatever we have done. So we know that for RNA-seq data workflow, we started from our data set selection. So we are going to select our data set, whatever data we need to work with. After that, we are going to download the FASTQ files. So these FASTQ files are the one that contains the sequencing data. Now these FASTQ files are going to be subjected to FASTQC, that is the quality control. Based on the quality of the base and the PCR duplication levels and all of that, we are going to understand from where we need to trim our data. If the quality of the bases are good and it does not have any bad quality bases, then generally trimming is skipped. And after that, if there are certain bad quality bases that are detected, then you go further ahead for trimming. So after you perform the trimming, then you align these trimmed FASTQ files with a reference genome. So we will have two files. One is a FASTA file and one will be a GTF file. So FASTA file is going to be a reference genome and GTF file is going to be your annotation genome. So you are going to map your particular reads onto your FASTA file, that is a reference genome, and check the alignment pattern. From that, further when you move on, you are going to calculate the raw count matrix. So before you directly align, you cannot just directly align your particular reads onto your reference genome. You need to index you need to index your reference genome before you align, because as the reference genome can be pretty large, so you need to break it down into small parts so that you can align it properly. So before you move on with the reference genome, so you first analyze and index the reference genome. So after you index it, then you can directly align your reads. So these are the tools. We can use Bowtie 2 as well. So here I would like to give you guys another for your project. So if you want to use any of these tools, Star and HiSat, and if your system supports, you can download these tools from on your terminal. And then instead of using Bowtie 2, you can use HiSat 2 or Star as an alignment as well. So maybe you can replicate the pipeline twice if you would like to use these tools, one with Bowtie 2 and one with HiSat 2 and Star. Like any of this you can apply. After that, when you get these particular files, if your SAM files are very larger in size, then you convert it into BAM, or you can directly process everything with SAM. For our data sets, we only processed it with SAM because we are SAM files but not that large. But I have also given the commands in the command sheet where you can convert the SAM file into BAM files. After you convert it into BAM file, you do sorting and indexing. All those commands have been mentioned. So if you would like to convert your SAM into BAM, you can follow those commands and perform the same analysis. So after you do the SAM to BAM conversion, then you do the gene quantification. That is based on this alignment pattern, you are going to count and calculate the read count matrix. So from here, you got the raw read count matrix. So this matrix is generated. So this matrix is now going to act as our input. So this input is going to be put in our HR or in a DEC2 analysis. So when you put it in that, then you can understand that which genes are differentially expressed or not. So there is an entire pipeline that we studied yesterday in HR where you're going to put, it is going to calculate the dispersion, the count matrix, the design matrix. Then it is going to tell how the experiment is basically formulated, and then it is going to calculate the statistical analysis test, that is p-value, log fold change, and all of that. Based on that, you are going to differentiate the genes into the upregulated genes and the downregulated genes. Using that, you plot volcano plot, PCA, heat map, and all of this plotting needs to be happened. Now in the process, we also downloaded certain like a file for genes. So those particular genes you can put onto online web servers like GeoEnrichment, Keck Pathways, and all of that, where you can get its biological interpretation. So this is the entire RNA-seq pipeline. So basically, if I have to sum up, RNA-seq is basically used to understand the expression differences that occur when you are studying two different conditions, either wild type or control, wild type or mutant, wild type treated, wild type untreated, whatever the conditions are, those are two contrasting conditions that you have, and you are trying to understand the expression level of the genes in both the conditions. If you have this particular goal, then the best thing to go is to understand and do the RNA-seq pipeline. So now I'll be talking about all the 7 data sets that I have given. So before I talk about this, first we'll understand. First, I'm going to allocate the data sets to all of you guys. So I know that all of you have access to this particular sheet. So here I have mentioned 7 data sets. So the first 2 are from Drosophila. Then next 4 are from Saccharomyces. Next 3 are from Saccharomyces cerevisiae. Next 2 are from Mus musculus. So Mus musculus is going to have a little larger size. So I just put 2 replicates here, but for others I've put 3 replicates. So now all of them, this is the control. This is the control section and this is the treated section. So these 2 are you going to compare and contrast. So now just to distribute, so we have 5 groups, right? Group 1, group 2, group 3, group 4 and group 5. So now the same thing will follow. So for the first data set, it will be given to group 1. Second data set is going to be group 2. Third data set is going to be group 3. Fourth is group 4. And fifth data set is going to be group 5. So now for each group, I hope there is a representative from each group. Okay, so like guys, can you give me a thumbs up that are you guys there? At least one member from each group. Okay. So based on this now, I have divided you guys into groups, 5 groups and the 5 data sets. So each group is going to access the particular data set. Okay. So for group 1, it is going to be the data set 1, that is on Drosophila melanogaster. For group 2, it is going to be data set 2. Similarly for all the 5 groups. Now here, I have mentioned these FASTQ files. So yeah, for the group 1, okay. So not just for group 1, for all the 5 groups, you will have total 6 FASTQ files. 3 are going to be control and 3 are going to be treated. So I haven't downloaded the data sets and given all of these are hyperlinked. So when you hover your mouse over one of the accession ID, you can click on the link that it shows below. It will directly take you onto the SRA website, where you all know how to download the FASTA and the FASTQ format. So from here you will download. So similarly, you will download it for all the 6 data sets here. Now I've also mentioned the GEO accession number. So you have to go to the GEO website. That is GEO. You have to go to the GEO website, and in GEO website, you are going to put this particular accession number. Okay, so here the accession number is this. So you will open something like this for an accession viewer. So you put the accession number here and you click on go. So now here you will get the study. So that is, the study was done on Drosophila. What is the study title? And what is the summary? Like basically you will get to understand that what basically this study is doing and what are the parameters that this particular study uses for what we got the data sets. So that is why I've given the GEO accession number, so you guys can study, and when you guys prepare your presentations and report, you need to mention a little about the study from which you took the data sets. So what parameters are these studies covering? So you mention that, you make people understand that what is happening, and that is when you took those data sets and you carried the work forward. Similarly, for both Drosophila and for Saccharomyces, it is there, the GEO accession number, so you can access all the data sets. All the FASTQ files have been hyperlinked, so you can directly hover over it and directly download the data sets. Is it clear? Okay. So now I have also decided the group. So now each group should very precisely and very attentively understand what I'm going to explain for each of it. And then we'll understand each and every data set in a little more detail. So now when we start with when we start with the data set 1. So data set 1 is basically covering the organism Drosophila, Drosophila melanogaster. Okay, it's a fruit fly. So this data set focuses on Drosophila melanogaster. Now the comparison here is happening between a blank RNAi that is acting as our control versus the CPS F73 RNAi. So it is basically a knockdown sample. So where you are knocking out one particular component. So what here is, what I want you to understand behind this study is that CPSF73. So this particular knockout that we are doing in Drosophila, it is a critical component of cleavage and polyadenylation machinery that is involved in the mRNA 3' end processing. So if mRNA 3' end processing needs to happen, then this should be present because it is going to cleave and do the polyadenylation. So in simple terms, if I have to explain, CPSF73 helps produce mature messenger mRNA molecules. So if this protein is depleted, the cell may experience widespread defects in the RNA mechanisms or RNA processing. That is, if CPSF73 is absent, that means the RNA is not going to be converted into a mature RNA. So therefore, it is very important. So this particular study aims to determine how this CPSF73 depletion alters the global gene expression. So the main biological question that we want to answer in this particular study, that is by data set 1, first is that which genes are becoming differentially expressed when we knock down the CPSF73, this particular knockdown. Second, how does this disruption of mRNA processing influence the cellular transcriptional program? So this is something based on the GEO thing. Like if you are going to disrupt this particular process, what further pathways are going to be disrupted? And when you are knocking out this particular gene, that means what expression is altering in between those control and the knockdown samples. So here in your data set, you have 2 samples, 3 blank RNA samples that are produced in replicates, 3 replicates, and similarly, the knockdown samples that have CPSF73 knockout that are also present in 3 replicates. So you're going to be processing that. Is it clear the theory behind the data set 1? So who and all have gotten the data set 1, can you please raise your hands? How many people are present from that group 1? Okay, so 2 people are present, right? Okay, 3 people are present, Maria, Navjeet and Girish. Okay, Varsha is not there today. Yeah, okay. Now you guys can put your hands down. So now we'll move on to the second data set. Second data set is also similar, where we are also working on the same Drosophila melanogaster. The study is same. You have the controls also the same as blank RNAi. There you are knocking out CPSF 3073, sorry. And now we are going to knock out the simplicin. So this is another thing that you are knocking out here that is going to affect certain processes. So this data set also uses Drosophila melanogaster. Now here the comparison that is happening is between the blank RNA, that is the control, versus the SIMP RNA, that is the SIMP knockout RNA. So what is simplicin? So simplicin is a scaffold protein that is involved in RNA processing and polyadenylation. So before it was here that it is involved in the polyadenylation machinery, which was present in the mRNA 3' end processing. Here it is involved in the mRNA processing and protein adenylation. So they both somewhat perform similar thing, but they're structurally a little different in function. So what is happening here is, like in CPSF73 that we just studied before, which was directly participating in the cleavage, so that mature mRNA is produced. Simplicin is not doing that. Here, simplicin is acting as a structural component that is helping in the assembling of the RNA complexes. Okay, so the major difference is this. So there it was directly getting involved. Here, simplicin is not directly getting involved. It is like acting as a protein that is a structural component which is involved in the processing of that particular process. So here the objective is to understand how this depletion of simplicin affects the transcriptome and the gene expression. So if we are going to reduce the scaffold protein, how is the expression level going to differ? So that is what we are trying to understand here. So the biological questions that we are going to ask in this particular study is that which genes are regulated by symplecin mediated RNA processing. So if in control, you will be able to find those genes that are highly regulated if symplecin is present, and in RNA, in the test sample where symplecin is deleted, you will be able to see that those genes are not that much expressed because symplecin is not only not present. So you will be able to detect these particular genes. And then the next question states that does knockdown of symplecin alter any specific cellular pathway? So once you understand the genes, again you put it in the gene ontology website where you can check and understand that are these genes that are getting knocked out and which are getting underexpressed in these conditions, are they leading to any problem during the cellular pathways? So you have to analyze it functionally, annotate it, and then you can understand that if there is any effect in the pathway or any enrichment effect that is happening. Is the data set too clear? Any doubts? Okay. Who and all are there in data set 2? Can you please raise your hands? Okay, Harita, Aisha, Surya, and any other? Gokul is not, no, Gokul is not there in the class today.
Gokul is here.
Ma'am,
Yeah, Gokul, you didn't raise your hands when I asked.
ma'am,
Yeah. Did you understand the data set that you have to work on? Do you have any questions, Gokul?
ma'am, I will come with later, ma'am.
Okay. Did you hear what I told about the data set? Do you want me to repeat?
Yes, ma'am.
Okay. So in the data set 2, what you are basically working on is you're going to work on with Drosophila melanogaster. So in this particular strain, the SIMP, that is a symplecin, which is acting as a scaffold protein, is absent. Okay, you are deleting that. So you have two samples. One is a blank control sample that has symplecin, but the other one is the sample that has symplecin knockout. So now what is symplecin? Symplecin is acting as a protein scaffold that is involved in the mRNA processing and polyadenylation. So in the previous data set, when I mentioned about in the previous data set, when I mentioned about the CPSF73, this was directly involved in the mRNA processing. But here it is not directly involved, rather it is providing as a structural component for this mRNA processing and polyadenylation. So therefore, you're depleting this symplecin and you're trying to understand if there are any complications or any expression level differences that are happening in the RNA. So the two biological questions that you need to answer here is that which genes will be regulated, which genes are basically regulating the symplecin mediated RNA processing. So once you do the knockout, you will be able to see when you compare it with the control that there will be certain genes that will be underexpressed or maybe overexpressed because they are not utilized as symplecin is removed. So you need to understand and analyze all those genes, and also you need to tell me that if this knockdown that is happening with symplecin, does it alter any cellular pathway, that is it is affecting any other pathway that is happening inside Drosophila? So that these two questions needs to be answered for this particular data set. Yes, Gokul?
Ma'am,
Yes, Gokul, go ahead, speak.
yes, ma'am.
Yes, Gokul, do you have any questions?
No, ma'am.
Did you understand?
Yes, ma'am.
Okay, so is your data now, this is the data set that you have to work with. Okay.
Yes, ma'am.
Okay, now you guys can put your hands down. So I hope it's clear the data set 2 now. So we'll move on to the data set 3. So now the data set 3 is in a different organism. Data set 3 is basically talking about the Saccharomyces cerevisiae. So Saccharomyces cerevisiae is affected, or basically it is put in some nutritional condition. So that has been studied here. So the comparison here is happening between the T0, that is the time like T0 versus 40 minute sulfur starvation. That is T0 was not starved for sulfur, whereas the other samples that were generated for T40, they were starved for sulfur for 40 minutes and then the sulfur was given. So now if we talk about sulfur in yeast. So sulfur is a very essential nutrient that is required for amino acid synthesis, for protein production, and for cellular metabolism in yeast. So when sulfur becomes limited, the cells rapidly adjust their gene expression to basically to survive. So this objective is to identify those genes that are responding during this particular starvation. Okay. So the two biological questions that you have to answer here is that which genes are activated or repressed within the 40 minutes of the sulfur starvation, and what are the earliest molecular response to the nutrient stress. So you will be able to understand that which genes are upregulated and downregulated. Based on that, you can determine the metabolism also, that are metabolism or pathway changes that are happening. So this particular study is basically telling that you have two samples. One is control, that is not sulfur starved yeast, whereas the other samples are those yeast that have been starved for 40 minutes for sulfur. And then you have to tell which genes are expressing more and which genes are expressing less. So here you are trying to understand the expression analysis in yeast during the sulfur starvation condition. Is it clear? Is the data set 3 clear? Any doubts? Who all have data set 3? Kindly raise your hand. Okay. Pravalika, Monica, Reena, and Dhanya. Okay, so all 4 of you are there. So is it clear to all the 4 of you? Okay, great. So now we'll move on to data set 5. No, sorry, data set 4. Do you have any question, Dhanya? You have raised your hand. Do you have any question?
No, ma'am.
Okay. So for data for set 4, it is similar to the data set 3. You're trying to understand the sulfur starvation response. Initially, you had 2 time points. One was at 0 time point and one was 40. Here we have increased the sulfur starvation. That is, we are going to starve the yeast for 80 hours. Not 80 hours, sorry. We're going to starve the yeast for 80 minutes, and then we are going to give the yeast the sulfur. So here the comparison is happening between the fully given sulfur yeast. Second is the yeast that has been starved for 80 minutes. The same thing you have to tell me what are the pathways that are getting altered, the expression level, that is the upregulation, downregulation, everything. The similar conditions like we did for T0 and T40. Here you have to understand for T0 and T80. Is it clear the data set 4, guys? Who and all are there in data set 4? Kindly just raise your hands. There are 3 people, right? Guru, Tanushri, and who else? Yasmin. Is Yasmin there with us today?
No, ma'am.
Okay, Tanushri. So are you guys clear with the data set? Okay. So now for data set 5, it is also similar, the starvation response that is happening in yeast, but here you are going to carry out the understanding for T0 and T120. So here the yeast has been starved for another 120 minutes. So now you should see a very significant difference that when you compare all these 3 reports. So first of all, for group 5, is it clear your data set that you have to compare between T0 and T120? Who are the 2 people in group 5? Are you there?
Yes,
Wafa and Hepzibah. Okay. So is it clear to both of you?
Yes, ma'am.
Okay, good. So now when all of you are done, then we are going to compare the genes. Like first of all, you have to compare within yourself, and then we are going to compare between these 3 conditions. So we will see from 40 to 120 which and all genes are getting affected more. Like in the initial, it may be not that much, but when you are increasing the starvation time, there will be overall effect that can be seen. So first you guys have to understand internally, then we are going to compare all of them together and also understand what is the trend that's happening. So these were the 5 data sets that was given to all of you. I've explained the objective and background behind them. I have also mentioned the accession number in the sheet. So you need to go through them also and understand once that what is the study about. So I would suggest you to read and then move forward. So I will explain the other 2 data sets also, that is for mouse. This is a slide that you need to a little avoid. This is, I have repeated one slide again. So mouse, there is two samples that I've given. So this data set is basically talking about the mus musculus. So this we are not giving to you right now. This, after you guys finish with the before data sets, this data set can be used to practice. Okay. So this data set is basically studying the gene expression change in the mouse mammary gland. Okay. So the comparison is happening between a virgin luminal cell and the lactating luminal cell. So one mouse is not lactating; it's a virgin mouse, whereas the other mouse is already lactating. So it is happening, and luminal cells are being compared between these two conditions. So during lactation, what is happening? The mammary cells will undergo major physiological and transcriptional changes to produce milk. So there will be certain genes that will be expressed more and expressed less compared to both of them. So what you have to identify here is those genes that are involved and that are involved in maintaining lactating. So when a mouse is lactating, there will be certain genes that are expressed. So that is why they're performing that function. But in the other part, that is in a virgin mouse, those genes are going to be suppressed because there is no lactation that is happening. So you will be able to identify those genes in mouse that are involved in the lactation process. So here you have to identify those particular genes and you have to establish that they are being maintained in lactation process. And then you have to also tell me the biological pathways that are getting activated. So for all the analysis that you're doing, first of all, you need to understand the highly expressed and the lowly expressed genes. Based on that factor, you will be able to understand which genes are involved. And after you understand the involvement of these genes, then you move on to the functional enrichment part where you are going to tell me the pathways in which it is involved. If it is deleted, then that means like what pathways are going to be getting affected. So all of that information is to be given properly. So similarly, next is another condition that is happening in mouse. So this data set is basically investigating the gene expression changes that is happening in a late pregnancy in mouse in the mammary basal cells. Mammary basal cells. Okay, so here they are both pregnant mouse and one is a virgin mouse. So initially we were checking in the luminal cells. Here we are going to check in the basal cells. So the comparison is happening between a virgin and a pregnant mouse. So pregnancy is going to induce extensive remodeling that is going to be involved in production of milk. So basal cells are play an important role in tissue organization and expansion. Okay, so there is some tissue expansion that is going to happen. So certain basal cells are going to be involved. So now when the mouse is pregnant, so those basal cells are going to be extending more as compared to a normal virgin mouse. So what expression level is changing so that the cell is expanding, that you need to understand from this particular study. So this is what the study is going to ask. The same outcomes is required, upregulation, downregulation genes, and which genes are basically expressed so that the basal cells expand. And similarly, what pathways and hormonal responses are there basically involved in. So now I just want to ask you guys, like, see, these are the 7 data sets that I have produced. So if any one of you want to perform on a different data set, like the first five data sets I've given, if you guys are interested in performing on 6th or 7th data set as a project, then you can suggest that as well. And if you guys want to take the 6 and 7 as a practice, you can do that as well. So whatever, it's fine. So if you guys are happy with the above 5 data sets, you can work with them. And if you want to change and choose the other data set to work right now, you can do as well. So is everybody okay with the data sets, or do you guys want to like choose the 6th or 7th as well? Or anybody wants to change, swap, switch, anything? Or are you guys okay with it? I don't see any thumbs up. So do you guys want to change or something? So everybody's okay, right? Okay. So now what I'm asking is further for the further studies. So what and all you guys have to perform. So each group should complete all these steps. First of all, you have to download your assigned RNA-seq datasets. Then you have to download your reference genome and download your annotation file. So for this particular studies, I have not provided the links for the FASTA file and the annotation files. So those two files you need to download on your own. So there are 5 groups. 2 groups are working on Drosophila melanogaster and 2 groups are working on Saccharomyces cerevisiae. So both of you have to download separately for the entire FASTA genome of melanogaster and Saccharomyces cerevisiae, and similarly the GTF file for both the organisms. So after you do the data acquisition, then you are going to perform the quality processing, where you are going to perform the FAST-QC alignment, construction of the read count matrix, and after that the differential expression. So while performing all these analysis, I would want you guys to create a doc. That is a log file. So give me 1 second. I'm going to show you, and I'm going to show you one example of a doc file. So you have to generate those file in a similar manner. So you need to write the commands. You need to write a small introduction. So all of that you need to write properly. Just give me 1 second. I'll show you that file. The time it's opening, yeah. So this is how it should look like, okay? So for RNA-seq analysis, there might be some formatting differences that might have happened. So it should write like this. You should have an experiment. Then you should have the name, aim of the experiment, principle in a very short span, what is the workflow including, and the entire procedure. So the procedure is going to be, you have to write the commands, you have to write a short comment over the command, and then you have to write the output of those commands. You have to paste the pictures. So here I have not added the inference, but you have to add the inference as well, what you are predicting and what you are understanding from these pictures that are getting generated. Similarly, you have to write the commands for all the steps that you're going to do. And then after doing the command, then whatever output you're getting, you need to write a small description of that. So similarly, the entire RNA-seq pipeline should be written with the commands. And when you get the plots, you need to put the plots. And here I've written the interpretation of the plots. So wherever there is a plot that requires, it requires a proper interpretation. So you need to write that interpretation for all the data. So this is how you should write, and everything should have an interpretation. All the commands should be included, and this is how your log file should look. It should look neat and clean, where all the commands can be seen properly. Is it clear? Can I expect this from all of you? Okay. So this is one log file that you need to generate, which has all the results. So you're going to be marked on the basis of all of this. Okay. So this and all I'll be expecting. So this is the assessment component that is basically there. So every step has been divided into particular part. So all these step marking will be based on the log file, because all the steps should be mentioned properly in the log file. If it is mentioned and accounted for, you will be getting marks for it. So the main marks is for the expression, the differential gene expression of data visualization and interpretation. So now your final, how does your final project looks? How is your final presentation? Your presentation skills, that will be accounting for another 20 marks. So the total weightage is going to be 100. That might be brought down for any short analysis, but this is going to be how the marking scheme is going to be. So you need to work accordingly and work accordingly, so that because you can be marked accordingly like this. This is the marking scheme. This is the weightage for whatever work that you're going to do. So now what and all I'm going to be expecting in your report, I just showed you. First of all, workflow documentation, the commands, the statistics, the count matrix, the differential expression results, the plots, PCA, heat map, volcano plots, then significant genes. This and all you will be saving in a file. The way we wrote in the file in our commands, the similar way you are going to write it in the file. Then you're going to find the functional interpretation. You're going to present all the work, and then you will be finally reporting it in a form of report. So all these things should be present in your report and final presentation that will be looked for, and that is going to be included in the assessment. So, and what and all in the end that I'm going to ask in the presentation is that each group should be able to explain how and why are the gene expression changing. So basically whatever I have mentioned under this biological questions, these questions needs to be addressed after you perform your analysis. For each and every group I have mentioned certain biological questions, so that needs to be properly explained. Is it clear how the marking assessment is going to be, what and all is needed, everything? Is it clear? Anything that requires an explanation? Okay. So this was all about your projects, guys. So these are the projects that you will be working on. And if you guys have any doubts, you can contact. I'll be available. So all the doubts will be solved. So we'll be having some doubt sessions in the middle as well. So you will be given 2-3 days to work, then again a doubt session. If you guys come across any doubts, you can directly contact, or you can wait for the doubt session as well. So this was your projects that was there. So I've also explained the projects that are here. Any doubts in the data sets, anything, you can just directly message. Is it fine, guys?
Anshu, I have one clarification here.
Yes, Pavani.
Regarding the reference genome, does they know how to download it?
Yeah, I have mentioned that before when I explained the FASTA sequence. So they can directly retrieve it from NCBI and save it in the FASTA format.
Can you please demonstrate it once now? I'm asking about the reference, not the GSC sample.
Yes, yes. So when you have to download the reference genome, what you have to do is you have to go to the NCBI website. Okay. So when you open the NCBI website, so you will go to NCBI website. This is the first one second, guys. So you will directly open NCBI. Okay. When you open this first website, you need to click on it. So say we need to have the Saccharomyces or Drosophila melanogaster. So I'm going to write Drosophila melanogaster. This is wrong. Yeah, it already automatically came. So now what we will do is we will search for nucleotides, because it is a data set that we want. So here nucleotide. Just let me confirm it once. I think there's some server error that is happening from the NCBI website, because that is why it is not able to search. Just give me a second. Okay. So here you can see Drosophila melanogaster. It's a cosmid clone. So this and all you don't need. You need the entire sequence. So you can see here the Drosophila melanogaster sequence, G007. This is this much long, 66, 168 base pair long. So you need to go through all of this, and you have to check that which particular section or which particular prompt is telling you the entire sequence. So for here time being, we're going to open this particular thing. So now this is showing that it has 6,000 bases. That is the DNA. So after going here, what you are going to do is you're going to click on FASTA. After you click on FASTA, an entire FASTA sequence file, like a FASTA sequence, will be opened. That will contain 66,000 base pairs. This entire thing. Okay. So this is your FASTA sequence. Now what you're going to do is you're going to download this FASTA sequence. So what you will do is send to. So what you will do here is you will do send to, say choose destination, file, FASTA format, and you will create a file. Okay. Now file has started to download. So you can see that the sequence.fasta has been stored in my file. So when I open it, okay, so this is the sequence.fasta that got saved. Now I'm going to open it with my notepad. Okay. So when I open it with my notepad, you can see the entire sequence has been stored. So this is what is going to act as your reference genome. So if you have to download the reference genome, this is how you are going to download the reference genome. For the Saccharomyces people, you have already worked on the Saccharomyces genome for your data set, like for the a classroom exercise. So it has both the data set files. So you can directly use it from there. But for the Drosophila melanogaster, you have to download it like this. So you have to find the entire file, and then you have to check that the entire sequence is there, and then you have to download in the similar format the way I have told. Is it clear? Okay. Yeah. Any other doubt, guys? Any doubts that you have regarding the downloading, processing, accessing, anything? Okay. If there are no doubts, then maybe this was it for today's session. I explained all the projects. Now these project lists are going to be sent. I have been already been shared with you. So after the session, you can directly start working on the projects. You can download the data sets. These data sets might take some time to download because they're little like they're big in size. So you have to start downloading, and you have to download 6 data sets for each person. So you can start doing that after the session. So now if you guys have no doubts and session is over, so maybe we can start with the assignment that I gave yesterday. Let me open the list of the students when all got what. So there are some people who were done with the assignments. That is, I'll just highlight them once. Okay, only the SRA people were done. So I'll just highlight them as red. Done. So now we'll start with Girish. So first we are going to start with the raw count matrix. Okay. So I asked everybody to do it. Did everybody complete that assignment? Did they do the calculations? Okay. So now it was Girish, Pravalika, Tanushri, and Harita. Are 4 of them present today? If 4 of them are present, can you please raise your hand? 1, 2, Tanushri, Girish, Harita, and Pravalika. Pravalika, are you there? Pravalika? Okay. So maybe we can start with Girish first. So Girish, can you present your screen and show me the calculations that you have done for the raw count matrix?
Yes, ma'am.
After
Sure.
Girish, it is going to be Pravalika, Tanushri, and Harita. So guys, be ready according to your turn. Yes, Girish, can you please present your screen? I'll stop presenting my screen now.
Yes, ma'am.
Yeah.
Girish, are you able to share your screen?
No, ma'am. I'm not able to share in the screen.
Can you please try now?
Yeah, now it's okay, ma'am. Ma'am, my screen is visible.
Yes, Girish, it's visible.
First, I will make a PPT, ma'am, for my presentation.
Oh, okay, okay.
I will done a calculation by using a R code, ma'am.
Okay.
Yeah.
That's good.
Yes, ma'am. So, first of all, what is a normalization? Normalization is to adjust the raw read count to remove the technical basis, enable accurate comparison of the
Can you
gene
share?
expression.
Sorry to interrupt you, Girish. Can you share
Sure.
your slide? Can you do it in a slideshow?
Yeah, yes, ma'am.
Yes.
Normalization is used to adjust the raw read count to remove the technical basis, enable to accurate comparison of the gene expression level. First, why we do a normalization? Our problem is the sample size is different, so we are unable to process the workflow. That's why only we do the normalization. By using a normalization, we corrected the sequence depth and gene length and library composition. In that, we do the three normalization steps. One is the RPM, read per million. It can be calculated by raw read count divided by library size divided by 1 lakh. And then RPKM. It is a read per kilo base million. It can be calculated by the RPM divided by gene length.
Okay.
First, we have to normalize the read depth by the RPM, and then we have to normalize for the gene length. And then last normalization is transcript per million. It can be calculated. First, we have to calculate the RPK. RPK is equal to count divided by length. And then we have to sum up the all RPKs. And then we have to find the scaling factor by total RPK divided by 1 lakh. And then and calculate the TPM by using the RPK divided by scaling factor. This is my working example. It can be given in the slide only. So I expect to differentiate the three normalization. RPM is used to depth only, not as for a length and cross sample. And RPKM is used to depth, length, not for a cross sample, but TPM is for all things. Now I will show my RPM code, ma'am. Yeah, this is a code. It is visible now.
Yes, it's visible. I was just saying that can you once go to the last slide?
Yeah, online. Last slide means summary or example.
Yeah, this
no?
one. Example. Okay.
Ah.
Okay. Yeah. Now you can go to your R code.
Yeah, okay. Yeah. First, I have to download the Excel file, ma'am. So, because I want Excel for the calculation after calculation, so I install the package of Excel and load it. And I create a data frame that is given by you, ma'am. And then I create a RPM calculation code, ma'am.
One
I will
second.
run.
Yeah, just let me see it once. Okay.
And then data frame. And then I load the samples. And then I will calculate the RPM values, ma'am.
Okay.
And then it RPKM and calculate for TPM as like a same formula, ma'am, in code. And then I creating a Excel file for my calculation, ma'am.
Okay. So can you show me the table that is created for this particular?
I will show.
That's a very
Creation.
Yeah.
After creation, it will stored in my documents.
Okay.
This is the Excel file. This is given by you.
Yes.
This is the RPM value.
Yeah.
After calculation done. This is a RPKM value.
One second. Stay on this. Just stay on this slide once. The RPKM slide.
Yes, ma'am.
Yeah. Okay. Correct. Yeah. Next.
It's a TPM, ma'am.
Yeah.
Yeah.
Yes.
This calculation should be more accurate. When we calculate by the calculator, some value should be differ from this calculation, ma'am.
Yes, I'm seeing some values that are differing from my calculation. So
Yeah,
just
yes,
give me a
ma'am.
second.
Yeah, yes, okay. Please check.
So just give me a second. Let me check it once.
Yes, ma'am.
So for your TPM values, okay. So
So
yeah, it's coming a little different.
don't rounding of the value, ma'am.
Yeah, that is what I'm
It's
just saying. Like my
38,
value is coming in
reason for
around
that.
1, 2, and your is coming for 1, 3. So
Yeah,
okay,
yes,
that's
ma'am.
correct. Yeah, it's just the
Yes,
rounding
ma'am.
of thing. Yeah.
I am also doing the same thing, ma'am. Manually it calculated, it showed the
Yeah.
deviation. It's
Yes.
don't rounding of the calculation, ma'am. That's only it's more. It should be more accurate.
Okay. Yeah. That's very good. You presented an entire R code. I'm very happy to see that the entire pipeline was run and you were able to give the values. That was very good. Very nice. Very good, Girish.
Yes, ma'am. Thank you.
Thank you.
I will stop my share.
Yeah, definitely. It was very good, Girish.
Thank you, ma'am.
Okay, so next is Pravalika, I guess. Is Pravalika there?
Sorry, Anshu, Pravalika is not in the call.
Okay. Tanushri?
Yes, ma'am.
Okay, Tanushri, share your screen once and show.
கோலை பத்தி எனக்கு என்ன வேல்யூல வந்து ரொம்ப அக்யூரேட் ஆகுது மேம் மேனுவலா கால்குலேட் பண்ணும்போது நல்லா வராது சிஸ்டம் கால்குலேட் பண்ணும்போது
Tanushri, are you able to share your screen? Yeah.
Ma'am, is my screen visible?
Yes, it's visible.
I did the calculation like this, ma'am, just as you presented them in the last session.
Okay.
Okay, ma'am.
So you calculated the library size and then you did the scaling factor. Yeah, correct.
Okay. Good evening, everyone. Today I'm going to present the normalization calculation performed on the given RNA-seq count data. I have calculated RPM, RPKM, and TPM normalization using the provided data set. So this is the raw count data matrix. So before starting the normalization, we can see that the raw count data and the total number of reads are different between the samples. For example, the control replicant 1 has a library size of 3277, whereas this treated replicant 2 has a library size of 5581. So since the total reads are different, so comparing the raw counts directly may not give accurate results. So therefore, normalization is required to make the gene expression values comparable across all samples. So first I'll explain RPM normalization. So in the raw count matrix, we have read counts for 10 genes across 4 samples, like the control replicant 1, control replicant 2, untreated replicant 1, and a treated replicant 2. So for RPM calculation, I first calculated the library size for each sample by adding all the raw counts within that sample. For example, the library size of this control replicant 1 is 3277, while this control replicant 2 is 3287. So like that, I calculated the library size for all the samples. So next, I calculated the scaling factors. So by using this formula, so here for this scaling factor, I used this library size and then I divided it in by 1 million. Then I got this scaling factor result. So next, so using this formula, I calculated the scaling factor for each sample. So after obtaining this scaling factor, RPM values were calculated using this formula, which is RPM is equal to read count divided by scaling factor. For that, I used this read counts and then I divided it by its corresponding scaling factor. Then I got this all RPM results. So each raw counts was divided by its corresponding scaling factor to obtain the this all RPM values. For example, for this gene which is YAL001C in the control replicant 1, the raw count was 125. So using the scaling factor of 0.003277, the RPM value obtained is 38144, 1.64449. So after calculating this RPM values for all genes, I proceeded with the RPKM normalization. So this is the calculation. So here the RPKM stands for reads per kilo base million. So RPM, usually RPM normalizes for sequencing depth, but longer genes naturally accumulate more reads than shorter genes. So therefore, RPKM additionally normalizes the data based on gene length. So this is the formula I used for this calculation. So here I used this RPM values, then I divided it by this gene length. So for example, the gene YAL001C has a length of 2 kb, and therefore its RPKM value in control replicate 1 was calculated by the RPM value by 2. Then I got this all the RPKM normalization results. So next, this normalization allows a fair comparison between genes of different length. So next, I performed the TPM normalization. So here, TPM stands for transcript per million. So this is the standard formula for TPM. So first, for TPM calculation, the first step is to calculate RPK values. So RPK value is calculated by using this RPK formula. I used this read count and then I divided it by its read length. So like that, I got this RPK values. So then, after calculating this RPK values for all genes, I summed all these RPK values within each sample to obtain the total RPK. Then I calculated the scaling factors using this scaling formula. So for this, I used this total RPK values and then I divided it by 1 million. Then I got the scaling factor result. Then finally, I calculated this TPM values by using this formula. So for that, I used this RPK values and then I divided it by this scaling factor. So using this formula, I calculated the TPM values for all genes across all samples. So these are all the calculation I did for this RPM and RPKM and TPM. So compared with this, raw counts, this RPM corrected for differences in sequencing depth, and RPKM further corrected for gene length, and TPM normalized the data in a way that allows easier comparison of gene expression levels across samples. So one observation from the normalized result is that this gene, which is YAL003W, showed consistently high expression across all samples in RPM and the RPKM normalization and also in the TPM calculations. On the other hand, this YAL010C showed very low expression values. So in control replicant 1, so the raw count for this gene was 0. So its RPM, RPKM, and the TPM values were also 0. So this indicates that no sequencing reads were mapped to this gene in that sample. So therefore, even after normalization, highly expressed genes remained highly expressed, while genes with very low or 0 read counts remained slow. Thank you, ma'am.
Thank you, Tanushri. That was a good presentation. I hope you guys are now clear with the how to calculate the RPKM and TPM and RPKM values. That was a good presentation, very nicely presented and good calculations. Okay, so now
Thank
we'll move. Thank you. So now we'll move on to the next. So next person is Harita. Harita, are you there?
ma'am.
Yes, Harita. Yeah, you can start.
The screen is visible, ma'am.
Yes, yes, it's visible.
Good evening to everyone. Today we'll see about RPM, RPKM, and TPM normalization. First, let's see about RPM. 1 minute. First, let's see about RPM calculation. This is the provider data of raw count matrix. This is the formula of RPM is equal to raw count divided by library size into 10 to the power 6. First, we need to calculate the library size. Library size means sum of all mapped reads in a sample. Like for sample 1, we need to calculate the control rep 1, all the values in the control rep 1 column. For each sample, we need to calculate the library size. It will vary. For control rep sample 1, the library size is 3277. And the second step is we need to calculate the RPM. RPM equal to read count divided by library size into 1 million. Read count is one raw counts, that is 125 for first read, divided by 3277. So we get the value of 0.038144644 into 1 million. So we get the value of 38144.6 of RPM value for first read. We need to do the same calculation for each read to get the RPM value for every read. This is the RPM value for every reads. Next, we need to calculate the RPKM, that is known as reads per kilobase million. This is the formula of RPKM, that is RPM divided by gene length. We already calculated the RPM value that is given before. So now we just update the values in here. So here RPKM for first read, RPM value for first read is 3701.6 divided by 2. So we get the RPKM value of 18507.5. This is the RPKM for first read. We need to do the same calculation for every read to get the RPKM value for each read. This is the RPKM value for each read. Then TPM calculation. TPM known as transcripts per million. This is the formula of TPM is equal to RPK divided by scaling factor. First, we need to calculate RPK and scaling factor. For RPK, this is the formula of RPK, raw count divided by gene length. Here, RPK raw count is 125 divided by gene length provided in the data. So divided by 2, so we get the RPK value of 62.5 for first read. We need to same the calculation for each read to get the RPK value for every read. This is the RPK value for each read. Next, we need to calculate the scaling factor.
For scaling factor, first we need to calculate the summation of RPK value. Summation of RPK means sum of all RPK values in a sample. This is the first we need to calculate summation of RPK for sample 1. We need to calculate the control rep 1 values of sum of all values in the column of control rep 1. So we get the summation of RPK value for sample 1. We need to do the same for each sample. So we get the summation of RPK value for sample 1 is 1377.26. Then we need to apply the value in scaling factor. Scaling factor is summation of RPK divided by 1 million. So we apply the values and get the scaling factor value is 0.00137726. So next step is we apply the RPK and scaling factor value in the TPM formula to get the TPM value. So here RPK value for first read is 62.5 and scaling factor value is 0.0013775. So dividing this value, we get the TPM value of 45379.5 for first read. We need to do the same calculation for each read to get the TPM value for each read. This is the TPM value for each read. That's all. Thank you.
Thank you, Harita. That was a good presentation. Nicely
Thank you.
explained as well.
Thank you, ma'am.
Okay. So I hope now everybody is clear with how are we calculating the RPM, RPKM and TPM values and how these normalization is happening, right? Okay, so now we'll move on to next. Guru, we can start with you. You can present your work on PCA.
Yes, ma'am.
Okay.
So screen visible, ma'am?
Yes, your screen is visible, but your voice is pretty low. Can you come again? Like, can you speak once again?
Can you hear me, ma'am?
Yeah, now it's clear. You can start.
Yes. So I will explain the PCS process in the RNA sequencing. PCA is a principal component analyzer, and it compresses the data into several principal components like PC1, PC2, and it reduces the entire data like in either 2D or 3D. In RNA sequencing, it typically measures the thousands of genes at a time, and it makes difficult for the visualization. So the PCA technique used here by dimensional reduction technique, and it summarizes the variations into similar number of principal components, which is the PC1 and PC2. And there's a cluster, means it's the plot, it occurs in the graphical representation. And the PC1, PC2 are the principal components. The graphical represent the individual frequencies, like the x-axis and y-axis of the graph. So this is the pathway of PCA. We have original data, like raw data of the multiple samples, which contains thousands of genes. Then it get compressed by the principal components, which each gene will be identified with a single number. And by the principal component algorithm, it will get into a single value of sample. Then it get viewed by the graphical representation. And the advantages of the PCA is to make the data set smaller, simpler, and easier to visualize. And it also have a disadvantage, which we cannot see the. If you only took PC1, PC2, the information from the PC3 cannot be seen there. So there is a disadvantage here. So there is a raw data, and the PC1 is the maximum variation from the sample, and the PC2 is the second maximum variation from the sample, which the information that cannot given by the PC1, and it gets compressed into a single score for a sample. Then it gets plotted by the PC plotter. So there is a clusters explanation. The samples are closer together, have similar overall gene expression patterns. So which are all the samples that get closer to each other have the similar gene expressions, and distance makes it dissimilar. And the PC1 is the x-axis, and the PC2 is the y-axis. It calculated by the variance in the algorithm, and the percentage of it gives the data information. Here in the graph, the S1, S2, S3 presented on the left, which is differentiated by the PC1, and by the PC2, the S1, S2 in the upper, the lower phase, the S3 and S5, S4 are the differentiated. Here by the commands, we first generating a normalized log PM values. It compresses the larger values to stabilize the variance by the values given by the gene expression. This is the example after the normalization happens, like a raw count is at 10, and the CPM value would be 3.3. It helps to compare the genes expressions, and here the transpose matrix. And from the value, it takes the input as rows and columns, like the samples and genes are included in this. So it centers the skills the data makes, and after this, the data can be comparable. So that here the data frame is created. By the data frame, it is created by the PCA value. The plot get visual by the graphical representation. Here is the final command, which is run in the RStudio to visualize the graph. Like this, the PCA plot, like PC1 and PC2 values are differed here. So these are the functions of the PCA, and this code, this is the final code. It takes the input of the PCA data frame, which we created at the last. Then scatters the plot. At each point, it represents the samples. The sample, which the number get by the PCA value, it get plotted as by the PC1 and PC2 axis. And here are the colors that are included to differ the samples, so that the PCA plot identify clusters, group separation, outliers, and the overall similarities at the difference in the gene expression data. Is it working? Hello.
Is it finished?
Yes, ma'am. Yes, ma'am.
Okay, great. Good presentation, Guru. I have just few questions. So initially, you like basically you explained the entire PCA based on the RNA-seq workflow, right?
Yes, ma'am. Yes, ma'am.
Okay. So I just want to know that did you check behind the math? Check the math that is behind PCA, like from where are we getting
Yes, ma'am.
those variance values?
Yes, ma'am. Yes, ma'am. Here you can see this, ma'am. The algorithm which is used by
Yes, yes.
The PC
In
one, ma'am.
RNA-seq, it's fine. Yeah. So in RNA-seq, we are basically taking the log CPM values, and from that we are
Yes, ma'am.
calculating, right? Yeah.
Yes, ma'am.
So in general, okay, before this question, I want to ask one more question. So can you plot PCA for any other computational analysis as well, or just RNA-seq?
We can plot for any other, ma'am.
Okay, okay. So the main function of PCA is to cluster. Okay, so I just
Yes, ma'am.
want to ask the same. Yeah, yeah. Yes, yes. Go ahead, Guru.
Here we can see the diagram, ma'am, the graphical representation, which defines like the gaps are the dissimilarities in gene expression, and the distance that close to each other are the similarities in gene expression.
Okay. So the same question I want to ask that, like, did you also go through the mathematical part that is happening behind the calculation of PCA?
Yes, ma'am. Yes, ma'am.
Yeah. So what are
Here
those?
we calculate the gene weight, ma'am. Firstly for PC1, if you have a gene expression, like commonly, if a person, taken as an example, we'll analyze the height and weight and etc. in PC1. And PC2, there is a different expression we are taking. Apart from the PC1 we took, here we taking another expression. This is called the second highest difference variation.
Okay. Yes, yes, that is correct. So what I was expecting as an answer, and like what I want you once to look into, is like the concept of eigenvector and eigenvalue. So that's a mathematical concept that is majorly circling around PCA and how those PCA's variations are calculated. So when you specifically do for RNA-seq, this is what is happening. That's absolutely correct and very well presented by you. But just I would want you to check once like about the eigenvector and eigenvalue as well, so that you can understand the PCA for the other concepts as well. So any other
Okay.
person has doubt? Are you all clear with what Guru has mentioned in the class? It is an important topic and he has very nicely explained what PCA is. So is it everybody clear with that? Can I get a thumbs up, please? Do you have any doubts? Okay. Yeah, Guru, you were saying something?
No, somebody else turned the icon.
Oh, okay, okay, yeah. So yeah, great presentation, Guru. Explained very nicely. I think everybody has understood because they give very big thumbs up. So I just want you to look into that eigenvector and eigenvalue once, so that you're thorough with PCA as well. For everyone, it is. Just look into that. That's a different concept of PCA. So is it clear, everyone? Okay. Yeah, nice presentation, Guru. Very well done.
Thank you.
Okay. Okay, so Bhavani, it's already 26. Should I start with the other set of people for that upregulation, downregulation experiment, or should we take it in the next session?
Shall I take it in the next session?
Yeah, yeah. Okay, then we can take that in the next session. Yeah.
Yes. Yeah, that would be fine.
Yeah, okay. So only those people are left. I think only how many people are left? 8 people are left that have to show upregulation and downregulation, and Pravalika is left for the raw count matrix. So all these 9 people are going to be presenting in the next session. So now as you people have your projects, so you will be working on that simultaneously. Next session is going to be a doubt session. So in that doubt session, first we'll take the presentations of the people who haven't performed today, and then we'll be carrying forward with the doubt sessions that are doubts that are related to your project. Is it clear, everybody? Okay. So do you want me to explain any project again? Anybody? I'm willing to, like, I'm ready to explain again if you guys want. If not, then maybe Bhavani, we can end the session for the day.
Okay, thank you, Anshu.
Thank you.
Group 1, anybody wants to ask any question? If your project is clear, please give thumbs up. Group 1. Thank you, Varsha. Thank you, Maria. Any offline students from group 1 is here? Group 2? Thank you, Surya. Thank you, Aisha. Thank you, Karika. Group 3? Tanya, Reina, great. Group 4? Fine. Group
Ma'am?
5? Yes, Varsha.
Ma'am, just one doubt, ma'am. In group project, we have two other like Anna University students, right, ma'am?
Yes.
We have to share our.
No, nothing is sharing here. Each and everyone wants to do it individually. We
Okay,
have
ma'am.
divided
Okay,
into group because Gene ID remains the same. In case if you have any doubts, maybe you can discuss. But when it comes to project, each one of you needs to work on it and submit report individually. It's not a group project. You're working as a group. That's it.
okay, ma'am. Okay, ma'am. Thank you, ma'am.
To your college also, you need to submit a report, right, question?
Yes, ma'am.
Yeah, it's an individual report. So each one of you wants to give us a final report. Okay. Please don't work as a group. Okay. If you have doubts, you can discuss.
Okay, ma'am.
Group 5. All right. Thank you, Varsha, for bringing this point here. And guys, please remember, this is not a group project, it's an individual project. If you have any doubts, maybe you can discuss with your group members. Plus, and if you are preparing any report, you can discuss. Please don't make it as a group project. Okay. Yeah. Anshu, do you want to say anything here?
No, I've already mentioned everything. So that's it.
Fine. Thank you, Anshu, and thank you, students. If you have any doubts, please post it in our WhatsApp group. And let's meet in our next session. I think it's on 18th or 19th. I'm not sure. Let me confirm with the schedule. And that will be a doubt clarification session. If you have any doubts, you can ask in that session. Thank you all. Thanks for joining. And please fill the feedback form. All the best, guys.
Yeah,
Thank you, ma'am.
everyone. Yeah, thank you everyone. All the best. So if you have any doubts, you can contact and then like just do the projects, like make a good presentation and the data sets that are given. If you find any problem anywhere, you can just post your doubts and there will be regular doubt sessions. So that's not an issue at all. Okay. All the best, guys.
Thank you, ma'am.
Thank you.
Thank you.